This method can help answer key questions in the peptide synthesis field, such as synthesizing disulfide-rich peptides and determining their corresponding disulfide-rich pattern. The main advantage of this technique is that it allows for the selective synthesis of different disulfide-bonded peptide isomers and their subsequent chemical and structural characterization.
Generally, individuals new to this method will struggle because each disulfide-rich peptide behaves differently. Synthesis and analysis may have to be partially optimized for an individual peptide or protein.
Transfer the reagents listed in the text protocol to the corresponding vessels and place them in the appropriate rackets of the solid-phase peptide synthesizer. Then, add 100 milligrams of dry resin to the reaction columns and put them in the racket of the synthesizer. Start the solid-phase peptide synthesis as detailed in the text protocol. After the synthesis is complete, lyophilize the resin overnight. Now, cleave the peptide from the resin and perform deprotection of the side chains. To do so, combine the dried resin in a 12 ml tube and cool it to 0 degrees Celsius on ice. First, add 150 microliters of a scavenger mixture. Then add 1 milliliter of 95% trifluoroacetic acid per 100 milligrams of resin. Leave the mixture gently shaking for 3 hours at room temperature. Now, filter the mixture through a glass frit. Collect the filtrate in tubes individually filled with ice-cold diethyl ether at 1 milliliter of